Thank you. So since we have a conference from evolutionary dynamics to active matter, my next slide is going to be about evolutionary dynamics. My last slide is going to be about active matter, and the rest is going to be about my work. My work is about microbial colonies, and I think this is a very nice system because it kind of combines both evolutionary dynamics and active matter. Again, as I said, the next slide is going to be about evolutionary dynamics, just to kind of convince you that microbial colonies are an amazing system to do that. So again, this is work from Roy Kishoni lab, and what they have is a colony, a petri dish the size of this table, and on the corners you have just the agar with nutrients, and in between you have the same pads but with increasingly high antibiotic concentration. And so over the course of about 10 days, you see evolution in action, with some maybe active matter in between, when the mutations appear, and then they allow them to grow at sort of 3 times the concentration of antibiotic that they would be able to do in liquid. And then, new mutations appear, and then they grow now at 300, or at 30 times the concentration, and eventually over here, they go at 300 and ultimately 3,000 times the concentration of antibiotic that was able to tolerate in liquid without any evolution. And you see that evolutionary dynamics is right there in your face, because you see the sectors of beneficial mutations appearing, taking over, competition between them, etc., etc. So again, this is not what I'm going to talk about. This is just to tell you that microbial colonies, especially the big ones, are very interesting. No, this is actual evolution. You can sort of study evolutionary-like processes by engineering specific strains and putting them together and observing a variety of spatial patterns. This is kind of indicative of evolutionary dynamics. So here, the first one, you just have 2 strains that grow at the same rate, but they have different colors, and demographic fluctuations lead to stochastic extinctions of one of the colors, and you have this characteristic sectoring pattern. So all of the noises people have been talking about, lead to things like that in microbial systems. The next one is an example of competition, where the black strain grows at a faster rate and sort of make these protruding sectors. You can have sort of mutualistic or cross-feeding interactions between them, in which case these sectors do not grow indefinitely, but sort of saturate to a finite length. And you can study other processes like horizontal gene transfer, etc. So these are sort of evolutionary processes that you can study in microbial colonies. And then, of course, microbial colonies for a long time have been known to be sort of a prime examples of active matter, where they produce a variety of patterns. And now, what I'm going to be telling you about is, this is all great, lots of interesting physics, but maybe too complicated for me. So I'm going to talk about, how this guy grows. A simple circular colony, and of course in this situation there is not much you need to describe the shape of this colony, just the radius, and then you can see how the radius increases as a function of time. Right? And in this particular case, for this Pseudomonas aeruginosa colony, the radius is a linear function of time, meaning that there's a constant velocity with which the colony grows. This is not unique to this microbe. People have, in a variety of systems, have found a fixed velocity of the expansion. And perhaps the most well-known and perhaps the first systematic study of microbial growth goes back to Pert, who have studied bacteria and fungi under different conditions, different nutrient concentrations, different plate sizes, different agar thickness, different agar concentration, and so have documented systematically how the velocity depends on all these parameters. And, supported by his data, he came to a conclusion that the growth is nutrient limited, meaning that, when you just put one cell on a petri dish, it grows exponentially. It just divides and divides and divides, and you have exponential growth. But at some point, that exponential growth has to be replaced by a slower linear expansion. And the idea is that, the cells grow, they consume the nutrients. Eventually, the nutrients get depleted, and they get depleted so much that only the cells at the periphery of the colony have access to the nutrients. Now I have a finite number of cells growing, and therefore the exponential growth is replaced by a linear growth. And I'm glad that Terry Wire came, because he was,
This is in a thin enough plate that doesn't have the bonded 3 dimensions.
Well, usually it's a standard microbial plate where you have either between 3 millimeters to 1 centimeter thickness of the agar plate. But the fact that the velocity is constant is sort of, you see it constant on a 3 millimeter plate and you see it on a 1 centimeter plate.
So does it cross over to being a constant later on the 1 centimeter plate?
So, you know, the PERT has some of those studies. You know, there's the agar does affect, you know, sometimes even the velocity of the expansion. Yeah, so that does play a role. But, you know, Terry Wa, Ned Ringren, who was here earlier, and Oscar and Bartolomei Walklow have, you know, perhaps other people as well, have put an alternative explanation for why the velocity is constant. And the idea is that instead of a nutrient limitation, they talk about some kind of a mechanical limitation. Let me give you a very hand-wavy explanation of what happens. So imagine, you know, you are at the edge of the colony where you have a monolayer of cells. Each of these cells elongates as it grows and pushes its neighbors. You know? And eventually at the front, you know, the cell, you know, is growing itself, but it's also being pushed by every cell behind it. So it kind of has a pretty high velocity. However, you know, as they push each other, the pressure accumulates, and at some point the pressure is so high that the cell loses contact with the substrate, becomes vertical, and at that point, you know, it kind of doesn't push horizontally, it just, you know, grows vertically. And so instead of a finite growing layer, you have a finite pushing layer. There's only a finite number of cells before the growth becomes vertical, and only these cells at the front are able to push. So again, you have a finite pushing layer, which eventually leads you to the same velocity, just the velocity is now controlled by mechanical parameters like the, you know, say, compressibility of these cells. Okay. So this is all great, but some organisms do not have a constant velocity. And particularly if you look at, you know, the classical, you know, lab strain of Bacillus subtilis, and so a variety of settings, it is the area of the colony that increases linearly with time, meaning that the radius grows as a square root of time. All right? And then the question is, what is special? Is B. subtilis special or E. coli special? So, you know, the best answer that I know comes again from Roy Kishoni lab, who, you know, took office scanners and converted them to measuring apparatus, where he was able to, you know, up to 500 colonies at a time, measure how the radius increases with time. Right? And so for a variety of soil isolates, you have a trajectories of the colony growth. And so generically what they found is that microbes are well described by a power law, and you have a distribution of exponents that characterize that power law. I mean, the linear growth, of course, corresponds to the exponent 1, and the square root growth corresponds to like 0.5, and so the median of the distribution is kind of close to 0.5, suggesting that perhaps the square root growth is the default in the microbial world if it was a representative sample. Questions?
There's a long period where
yeah.
measure, but it's sort of exponential.
No, it is a lag. It's a lag time, right? Usually, when you grow them in liquid, and then put them in a petri dish.
But this is a lag time of 50 hours.
For some organisms, yeah. Right? We don't know because again, this is not E. coli that we have, you know, represented all the time. So this is natural isolates, and you know, perhaps some of them don't even grow at all on the.
The lag time is also the time for it to grow to an exponential region and for it to slow down. Which also will be.
Somewhere here is an exponential region, right? So you know, I don't know, like whether, you know, I presume that most of that 50 hours is a lag phase, and then you have some short exponential phase, and then you have the other growth, right? Because you can see like these guys grow at a comparable rate, but this takes 100 hours. This takes 50 hours. So presumably most of it is a lag time. Okay, so here, yeah, question.
Maybe I confused or something, but I think this 50 hours is how long it takes for the colony to be visible, because it starts with it may not start with one cell, but I would say out of that, 150 hours, some of that time is the lag time, and I think the lag time is the most of the time. Right? I know that for E. coli colonies, it takes up to 24 hours to switch from sort of exponential growth to linear growth. Yeah.
5, 6, 7.
Yeah.
This time space is short enough. No, it's long lag.
change is the expansion of the radius. You're talking about like 0.5 millimeter in a couple hundred hours. That's, what kind of a, I mean, are these, you know what kind of bugs it is?
Oh, it's a soil, it's a natural soil isolates.
What?
Soil isolates, natural. So it's up to 500 different organisms. It's not lab strains. Yeah. Okay, so this is nice, right? Yeah.
I mean, if you took a 3D blob of spongy stuff and inoculated the center of it and supplied with everything it needed, would it grow as a volume growth or something else?
Well, that's a good question. You know, I mean, obviously these experiments don't answer that, right? Right? And I guess I would say, you know, we'll talk about it, right? You know, and the models, of course, can be generalized to 3 dimensions, but it's a very obvious question for tumors. And again, there are a variety of, you know, very special so-called gumball growth curves for tumors, which are, you know, neither here nor there.
If I take it, the sensitivity here is, you know, 100 micron scale, something like that. So I have there's quite a bit of exponential if it's growing every few hours before.
These definitely, exponential has to be there, right? Because, you know, when you have a small number of cells, they do grow exponentially. And then it switches, but, no, again, what we observe is a non-exponential growth, and that's what we want to understand. All right. So this is where sort of experiments are, where the theory is, you know, I guess it's a, you know, on a simple approach. So we're going to start kind of, well, from, you know, far away with the phenomenological models of growth, right? Not the mechanistic models. You know, the mechanism is important. We just want to understand, you know, is there a phenomenological model that, for example, recapitulates this behavior, although we will at some point go to the mechanism as well. But we start with the phenomenological level. So the simplest phenomenological model of growth is the Fisher equation that people have introduced before. Again, it combines the regular diffusion of the biomass, B, which is bacterial biomass, and logistic growth, which captures exponential growth initially and then saturation and some carrying capacity. If you put this thing on in space, then you end up, you know, first you have local growth to the carrying capacity, and then the outward expansion with the fixed velocity v that is given by a combination of the diffusion and growth. Right? That's a standard result. You know, this equation is clearly not applicable to microbial colonies I showed you before, in part because, you know, they don't diffuse, and this equation has exponential tails which do not occur in microbial colonies that typically end up with a single cell at the end. However, this equation does apply to some microbial colonies in particular. Well, I mean, it could apply to some microbial colonies. It was argued that it's applied to some microbial colonies. So this paper by Wakita et al. Have studied bacteria in a soft agar, where they're actually swimming, not pushing on each other. And in this case, they've measured the growth rate as a function of nutrient, which is, of course, linear in nutrient concentration. They measured the diffusion constant as a function of nutrient. Again, this is the colony, this is the center, this is the periphery, so you see that there's a gradient in density. But, you know, the diffusion constant does not depend on the nutrient concentration, and therefore you expect that the velocity, which is the square root of d times r, is going to be square root of the nutrient concentration. And indeed, that's what they found, you know, by fitting v as a function of nutrient concentration. Okay. So it just was, you know, we kind of got sidetracked from our colonies. You know, but the Fisher equation always predicts you a linear expansion, right, with a constant velocity. So then the following model became quite popular, trying to actually understand the colonies on hard agar, where the cells are pushing each other. Right? It goes under a variety of names of a nonlinear diffusion population pressure model. So basically the key difference is that now our diffusivity is density dependent. And so we'll talk about the origin of this maybe more carefully in a couple of slides, but, you know, very qualitatively you can say, well, if the cells are pushing on each other, if we are kind of sitting sparsely and try to push, we won't going to be very successful. However, if we combine, say, in a tiny office, then perhaps, you know, we can push each other much more successfully, and, you know, that's the origin of this beam, right? Okay, so the key reason why people have sort of latched onto this model is that in addition to having a sort of a velocity, it predicts a profile which is very different from the Fisher equation. Here, the profile does not have any exponential tails. It sort of goes to carrying capacity at the back, and then the density vanishes completely at a certain point. Right? So that is very much like we have in a microbial colony. So that's good.
And
What happens if you introduce a cutoff? What happens if you introduce a cutoff? Because B will, no, here your diffusivity is falling to 0 when B falls to 0, yeah. But actually when the diffusivity falls to 0, it should stop at the single cell diffusivity, which is finite.
Well, so, because here the density vanishes, you don't have to have a cutoff. You could have added a cutoff in the Fisher equation and say the diffusivity vanishes below a cutoff, right? Then you would end up with an equation like this, but this is somehow emerged naturally, right? So, you could, again, you can put an arbitrary power of B over here, and then depending on the power, you can approximate a cutoff better or worse, right? So again, it's your choice whether you want to do a cutoff versus you want to do a power.
Yeah, what I meant is if when B goes below some threshold, this becomes a constant, then you would be locally linearly unstable and you would have the same Fisher selection mechanism. And I'm just wondering how these two speed would.
So if you put a cutoff over here, you will end up with a Fisher tail. But this, obviously, we kind of chose this model to avoid the Fisher tail. So that's why we're not going to add a cutoff over here, right? And also, in a real microbial colony, right, if you reach one cell density, then you're going to replace this D not by a constant, but by 0.
But then you have this log correction,
no, those guys, they correct the growth rate, not the diffusion. All right. But despite all of this interesting discussion, the long story is that with or without the cutoff, there is a constant velocity. We still don't have our square root growth. All right. Okay. So before let's connect with the soft matter, right? So this model, is known as a population pressure model. Again, the population pressure the name population pressure comes from ecology, where population pressure means that you don't like higher density. For example, you go to high density, you say, oh, this is not great, I'm moving to Alaska, right? So this is the origin of the name of the model, right? It was applied to insects where that was indeed the case. Now, turns out that this model can be derived from a physics-based pressure, right? So now if we think about these cells pushing onto each other, and we can write down a simple conservation law where there's the growth, that's one way to change the biomass, and then there is the transport of the biomass with some velocity, right? Clearly, velocity comes from pushing, and there's a balance between the pushing force, which is the gradient of pressure, and the friction with the substrate. Now, to close these two equations, we have to somehow relate pressure back to the biomass, and there's nothing simpler than a linear relationship that you can always justify by Taylor's expansion. Right? And if you now combine these three things, you do end up with the equation that I had on the previous slide. So with the diffusion constant given by the ratio of sort of some kind of compressibility and the friction coefficient. Again, pocket this into your mind. We'll come back to it at the end when we actually discover an area law rather than a constant velocity. So again, coming back is that we still haven't seen anything but a constant velocity. And perhaps we now go to a different model because the square root growth somehow may think about like a nutrient depletion, etc. So you may say, well, let me add the nutrient dynamics explicitly. So here, N is the nutrient concentration, and gamma Bn is the growth rate, and at the same time is the consumption of the nutrient because we chose to measure the biomass and the nutrients in the same units. Right? The nutrient and the biomass have different diffusivities, and so that's the model. This model was very popular, became very popular, because it explained the following phase diagram. So this is an experimental diagram, this is some simulations. The idea is that if you vary the nutrient concentration and the agar concentration, then the patterns that you observe in the petri dish are very different. And this model was able to reproduce that by varying the biomass diffusivity and the nutrient concentration in the media. However, this model was carefully studied by Van Sarles and Muller as far as the velocity goes. And what they found is that whatever you do in this model, it always has a constant velocity, although the velocity now has a slightly different dependence on this parameter d. Right? Instead of a square root, you get a linear dependence in some regime. So I think now it's time to sort of actually tell you how to get a square root growth, right?
And so we have to make a very simple change, right? So what we're going to do is we're going to introduce nutrient concentration over here where the sort of nonlinear motility goes, right? So you can call this model a nutrient-dependent motility. However, that's mathematically accurate because in the motility we have the nutrient. But I would say the proper way to think about it is a growth-dependent motility, because this term Bn is the same as the growth rate. And I'm not telling you that the cells on a petri dish sense the nutrient concentration based on that adjust their pushing level. What I'm trying to argue is that the growth rate of bacteria affects somehow the efficacy of pushing, right? I don't have any sort of direct evidence for this, but I have some indirect evidence that maybe have motivated this model, and that again comes from these competition experiments. So here in that study, the yellow strain was susceptible to antibiotic, the black one was resistant, and therefore by tuning the amount of antibiotic in the media, you could have varied the ratio of their growth rates and sort of the ratio of the expansion velocities that determine these sort of sector shapes. And what was found is that the velocity was proportional to the growth rate, not to the square root of the growth rate, which you would expect if the motility was not did not depend on nutrient concentration. Okay. There was some indirect evidence that perhaps the motility should depend on the growth rate, but for quite some time, let us just maybe put on mathematical hats. We have our model. Let's figure out what it predicts. Okay. So to do that, we non-dimensionalize everything, and basically our model ends up with just a single parameter d, which is given by the ratio of the biomass motility, the nutrient diffusion constant, and the nutrient concentration. Right? And that's what we're going to be varying to see what happens in this model. So the long story short is that this model does predict both the linear expansion and the square root growth. So for different values of this parameter d, we plot the total biomass against a one-dimensional simulation. So the biomass is the same as like position, more or less. And what we see is that sometimes we have a linear expansion, and sometimes we have a square root expansion. Right? You can systematically vary the parameter d and try to fit either a linear dependence or a square root dependence. And what you find is that when this biomass diffusion is low, the square root fits better. When the biomass diffusion is high, the linear expansion fits better. So it looks like we have a transition at some value between these two different regimes. So now, let me just maybe briefly go through what happens in each of these regimes, and then maybe come back to biology and active matter. So let's start with the traveling fronts, right, with the constant velocity. Again, nothing weird, right? We have the biomass profile, which is very sharp. The nutrient profile kind of about the same width, and they follow each other sort of at the same velocity. You can sort of measure the velocity as a function of that biomass parameter. So this plot has a bunch of stuff, so let me kind of walk you through that. So the dots are the solutions of the PDE. So I solved the PDE for two different system sizes. When I do have an actual velocity, the two system sizes give me the same answer, and that's the velocity. Now, when I don't have a velocity because I'm in a square root regime, the bigger the system size, the smaller the velocity, and eventually it vanishes completely, right? The dashed line is a solution of a shooting method for the ODE, and the black line is sort of a rough theoretical calculation. In the interest of time, we may not go too much into the calculation, but basically, if you make an ansatz that you are moving with a fixed velocity, you substitute that into your differential equation, you end up with a system of ordinary differential equation, which you can analyze by asymptotic matching, and you can kind of find an approximate answer for the velocity. So at large d goes as a square root of d, it vanishes at d equals 1, and it vanishes linearly in this parameter. Right? And so that's what was shown there, approximately. Now, in this other more interesting regime, we have many differences, right? So first of all, the front is slowing down over time. Then there is a very large depletion layer ahead of the front. There's almost no penetration of nutrient behind the front. Also, the thickness of the colony is now higher than 1 because it absorbs not only the nutrients over here, but also some nutrients ahead of the colony. And the front itself is very sharp, at least compared to the nutrient profile.
This is the same equation.
The same equation, yeah, just a different regime, just a different value of the parameter. And so we can analyze this regime by making the following ansatz, is that the biomass is just some height times a step function which moves with some rate. And for now, let's just assume that we know h. We will first solve assuming h is known, and then we're going to find h. So if you do that and you have this ansatz, you basically have to solve for the nutrient dynamics. That is solved, using solve similar solutions, and eventually you can find the nutrient profile, and you can find an equation that relates height, which we assume we know, to the rate at which the population grows, right? So this var kappa is a measure at which it's not a velocity, it's basically the rate at which area grows. And you can find that through this equation. I mean, this does depend on dimensionality, so again, I will talk dimensionality about later, but you can solve it in any number of dimensions. The answer is a little bit different, but that's not super important. Qualitatively, what happens is that if you have a slow expanding colonies, then from this equation you can find that the height is going to be like 1 over kappa, it's going to be large, while if the colony is fast, then the height is approximately 1, right? So, which is the same as in the traveling front limit. Finding h is a little bit more involved. Again, it's some kind of asymptotic matching. But if you look at this equation, you try to do some math, assuming you have a slow varying velocity, you can eventually find that h is 1 over square root of d. And so this is a numerical check of that. So this is the thickness of the colony as a function of the biomass diffusion from PDE, and we have a very good agreement. Now, if you combine this expression for h with our expression for the var kappa before, then you can predict the expansion coefficient kappa as a function of the diffusivity. And again, you have a very good agreement between the theory and the simulations. You do see that you move faster in 2 dimensions because you have more nutrient. That's not me. I guess it's an important point we're trying to make over here. Maybe it's a microphone. If it's my computer, no, my computer is now muted.
Yes,
And that doesn't make a huge difference.
This doesn't make any difference.
Okay. Something made a difference. Okay. So. Okay. The most, one theorist can fix a projector, right? Okay. So the dimensionality does play some role. In a sense, in this regime, when you have very low diffusivity, you actually have a very different behavior of the expansion rate as a function of nutrient concentration and other parameters. And that's perhaps the easiest thing to test experimentally, because you can grow a colony in a channel, and you can grow a colony in a petri dish at different nutrient concentrations, and you can check for linear versus square root dependence. Now, one thing I want to emphasize is that this sort of regime that we have is very different from a stationary absorber, right? So, if we think about a colony that doesn't move anywhere, just sits in one place, the nutrients still come into it by diffusion, and its total biomass is going to grow over time, right? So that limit can also be solved, and that's the dashed line over here. However, if you have some biomass motility and you still grow as T to 1/2, you move at a much faster rate, right? So it's not the same. So this second regime that we have in this PDE is not the same as a colony just sitting and absorbing the nutrients. And now, if you solve the simple model, you can of course say, well, maybe I can get, so here we have only T to 1/2 and T to power 1, right? If you want to get other parallels, perhaps you can do a crocodile like this. We looked at it very briefly, and basically we found that the key parameter is this exponent alpha, such that when alpha is less than 1, you only have the velocity. Alpha equals 1 is the case I've talked about most of this time.
And when alpha is bigger than 1, you again have two regimes, a linear regime and a different regime, but now there's some subtleties. So the biomass always goes as t to a half, but now the position would go with an exponent less than a half, because not only the colony is going to grow horizontal, it's also going to thicken as a function of time. This is sort of illustration of that. Now the different colors are different values of alpha, and the biomass they either go to 1 or 1/2. The positions now have a broader range from 1, 1/2, and less than 1/2. Again, we still don't have anything between 1/2 and 1 asymptotically. However, if you look at not this parameter 1, which is close to transition, but as a smaller value, not that much smaller, right? Say 0.2. Now you have all very long transients. And these sort of limiting cases where you're supposed to get exponent 1 on a sort of short time scale, short meaning up to 10 to the 5, they look like a power law. And so perhaps, not asymptotically, but on a practical level, this model could be consistent with the observations by Kishoni of a variety of power laws between 0 and I would say, I'm not sure about things that are bigger than 1. Right? So to summarize, I have told you about some experiments about different growth laws. We proposed the phenomenological model that I think explains at least some of them. And when I end is with some questions. Now, first of all, of course, we might have put some more, interest in the debate between mechanics and nutrient diffusion, because our model is primarily based on the nutrient. But I would say the more interesting question to me is this one, which connects with the soft matter. So if we think that the motility has to depend on the growth rate through the then how does that happen in this mechanical calculation? Right? The mechanical calculation is based on some ground truths like biomass conservation and friction, which I think we believe in. So this ratio of compressibility and friction has to somehow depend on the growth. And in our classical mechanics, right, compressibility is just, we have these, spherocylinders in the simulation or something, they're just mechanically compressed. But perhaps if you have a growing matter, then there's some additional active stresses that one has to think about that would make this dependent on the growth rate. And that's not a statement, that's a question that I, hope to discuss with any active matter people, because to me, I think, it's not clear that the compressibility of an active material would be independent of its growth rate. I think generically there should be some dependence, but I don't think it has been studied. Maybe it has, but I don't know about it.
This term does look like what you get in hydrology.
Have what?
What you get in hydrology.
Hydrology? Well, I'm not in hydrology.
Squared instead of then you get a p squared cavity.
Talking about this term?
That term, yeah.
Yeah. So you can get different powers of B. There's no problem with that. But now you have a dependence on n, which is dependence on the growth rate. Right? I don't think that happens in hydrology, right? So it only happens in active matter, because the idea is that this thing does not produce you a square root growth. This only gives you a linear growth. So to get a square root growth, this thing has to depend on n. Of course, n is like really the placeholder for growth, which means that your perhaps compressibility should depend on the rate at which you grow. Right? And then the question is whether it's true in any active motor system. Yeah, we're done. It's time for questions. Well, this is my question to you. And if people want to make a comment on this, we can start the discussion.
Go ahead.
Okay. Thank you for the interesting talk. So if I understand correctly, there's a sharp transition in D from T to the 1/2 to T to the 1. I'm curious do you think if you had noise, demographic noise, that would still be the case? There would still be a sharp transition? I'm just wondering if like the DP transition and near extinction would be different on either side of this phase of this D boundary, but maybe that's not relevant. I don't know.
Yeah, I think here there will be no major effect of demographic noise because it's really about the nutrient. I mean, the way you can think about it conceptually is that if your motility doesn't. So let's think about the situation where alpha is 0. You only have dependence on the biomass, right? What happens as you sort of make this parameter d smaller and smaller? You have a larger and larger depletion region,
Therefore, the nutrient concentration at the front gets smaller and smaller, and your velocity gets smaller. But you never, like, you know, you always can keep up, sort of to speak. Now, when this thing depends on n, you have this runaway process, right? You move slowly, you have a larger depletion region, your nutrient concentration drops, and you move even slower. And so that sort of positive feedback leads to a transition where you cannot maintain a constant velocity. Any type of fluctuation in the density at the front is not going to really make any difference. It's really about the nutrient dynamics, which happens over very long scales, and this, you know, averages out all the fluctuations.
Somewhat relatedly, if you had both kinds of diffusion term, this one and the old one, what would you get? Would you always get linear?
I mean the
regular diffusion.
Well, if you have this one, right, and just a regular diffusion, right, then of course this is going to go to 0.
Right.
The regular diffusion survives,
okay.
And then of course you move it with constant
velocity. So something has to forbid that other term, like that.
Yeah, you have to forbid that other term, but again, that term is forbidden because it expands on hard agar. The word hard agar means that you can't swim.
Okay.
Yeah.
Thank you.
Is it important that the growth term and the nutrient consumption term have the same functional form? Because independent of the coefficients, the fact that you have p, n in both of them means that always one linear combination of b and n obeys the conservation law. Yeah. So and that's not generic, right? Why did it have to?
So let's go to maybe a bigger equation. You know.
No, not there. I'm saying the last, the gamma, your b to the mu n to the nu outside.
This is the most general equation, right? All the equations I have studied are.
No, it's fine. I'm just saying the part I'm talking about is the fact that you have a b to the mu n to the nu in both.
So I'm telling you, the exponent beta makes no difference.
Right.
Yeah. So you can have the fact, so for example, in the previous model with alpha equals to 1, I can have using beta equals to...
That's not my question. My point is because the terms without gradients, that term appears in the same form in both equations. Means one linear combination of b and n always obeys a conservation law.
Yes, so this is a conservation law because it means that if you eat a nutrient, that nutrient becomes your biomass. You don't waste the nutrient. And I basically have rescaled my units of the biomass in the nutrient. So typically in biology we would have so-called a yield coefficient that tells you how much of a gram of nutrient makes a gram of the biomass. I rescaled it to make it 1. Yeah. You could have other situations where you have like a maintenance cost, right? When you have a maintenance cost, when you basically some of the nutrients just basically used as energy rather than as a constructive material, then you have a situation where at some point you're going to move as an area law. And at some point, the nutrient is going to be so low that it's only enough to maintain you, and you're going to stop growing overall. So it's going to have growth and then saturation. Nice. Right? So again, it changes to asymptotics, but it is consistent with real life because you have some growth and then you do saturate.
Nice.
Okay. So in order to generate the theory with this variable alpha, what kind of microscopic model could you formulate?
Okay, so let's go back to the end. That's my mechanics argument. So this is these microscopic models eventually do generate anything like this, right? So if you make any power law here, you're going to have another power law over there, right? The only thing that is not clear about the nutrient is how these guys depend on the nutrient. And in the standard derivations, people assume that they're independent of the growth rate. And I'm sort of saying that perhaps that's not justified, or it's not clear to me why that is done, right? So that's a microscopic model. I mean, maybe not as microscopic as you want, right? Because maybe that's a question then to you, right? If we take a microscopic model of cells, right, of like really this granular material that is growing, whether that granular material, its mechanical properties should include some dependence on the growth rate, right? So, yeah. I hope that answers the question. This may be. Yeah.
What happens if your nutrient is biotic and grows itself? Do you get Turing patterns, or because it sounds like that's the limit where nutrient diffuses fast and faster than the.
Yeah. So in a microbial colony, the nutrient, fortunately, does not divide Again, if you have sort of predator-prey effective dynamics, then you could have different oscillations, patterns, etc. I think all of those studies have been done in a situation when the motility of the predator does not depend on the prey density. All right. So I guess we're asking that if motility depends on the prey density, you may have new phenomena which would be not linear expansions, but the square root expansions. And as far as pattern formations, how that affects, we of course, I have nothing to say about that, but there could be some changes.
But you think they could combine so you would have sublinear growth of the colony and something like Turing patterns happening together?
I see no reason why not. Let's put it.
Thanks.
Given that we haven't done any work on that.
Okay. I have comments in several parts.
Yeah.
First of all, about the theoretical description, right? So I think at least for our system that we look at the E. coli, I don't believe in the Fisher equation.
No, of course not.
Right?
This was clear, right?
Yeah. And you try to invoke the Fisher equation by first talking about some dense colony, swimming, and that sort of thing.
That was
but you're still applying it to actual growth on plate.
No. So
right. I mean, eventually the square root comes from the growth on plate.
Yes. So
you try to apply to growth on, but there.
So the only time this this Fisher equation came was more like historical introduction.
No. I mean this type of a reaction diffusion equation.
Oh yes, reaction diffusion. Yeah, it's a reaction diffusion equation.
Okay, because basically, I mean, it's not clear what diffusion or motility means, because these things, if you look into the column, they're completely frozen. I mean, even the periphery. They don't change at all. They're completely locked in.
What do you mean? These sectors definitely move.
Even just the orientation of the cells and so forth, they're completely locked in. Okay, and they're growing in the periphery cells. They're growing exactly at the same rate, and you can change nutrient concentration. Nothing changes. Right? But I'm of course applying to. I'm talking about the system we're looking at.
Yeah, E. coli.
Right? Yeah, but then. Okay, so I was. Very surprised by the result that they showed with the Kishoni's result. Yeah. Right? What struck me was the colony size was so small. Okay, and then so I looked into the method just now.
Yeah.
Okay, and you'd be surprised.
Yeah.
In the experiment, they put extremely low concentration of nutrient.
Well, or you put a very high concentration.
I mean, not me. Typically, so what's common, right? So typically, people when you ask
in a lab, right? But again,
you put lots of nutrient.
Yeah.
Right, and so they're just limited. That's where you see the limitation by the physical force. The nutrient is absolutely not a limitation. But in this experiment, they put such a low and they tell you either in a method, right? And then I look at the number, I calculate the concentration is abysmal.
Yeah.
Okay. So basically. My interpretation of their result is they're simply running out of nutrient, and you're just seeing the saturation and find somewhere in between you fit it to a square root.
Well, first of all, simply.
No, there's nothing to defend. That's just the experiment.
It's good. I just make a comment on your comment, right? Yeah. Because there are two things, right? First of all,
you may still be right, but I'm just saying.
No, mathematics is probably right.
Don't take it seriously.
One thing is that about the. Yeah, so the nutrient limitation by itself, without motility depending on the growth rate, would not give you a square root. It would only give you a slower linear.
You get some kind of classical. We looked at concentration dependent on these things. Of course, you get sublinear growth. It slows down.
It eventually slows down. But again, these plots.
Eventually, it just happens much larger for our scale.
These plots slow down from the very beginning.
This is extremely slow. This is extremely low nutrient concentration.
And now you have. Bacillus subtilis at 2% glucose that shows.
Bacillus subtilis is completely different. If you look at the Bacillus subtilis, they're expanding huge, extremely fast. Because they're secreting surfactant, completely different game.
Well,
okay, so you got to look at numbers, not just look at the power. I mean, I came from a power law education, but you need to look at numbers.
I tried to bash it out.
So surfactants are, I know, Bacillus has a different mechanism, but again, at very high glucose, it has this power law diffusivity, right? And then,
I didn't get your point about Bacillus, because Bacillus is swimming.
No.
They're excreting a lot of surfactant. Right? So they're swimming. I mean,
at 6% agar, Bacillus is not swimming.
Huh?
At 6%, agar B cellulose is not.
6%, okay.
Yeah, no, the 6% right here. 3 is 1.5, right? So again, this is not swarming, and this is not what you have like 24-hour covering the petri dish, which you have like when Pseudomonas aeruginosa swarms. This is still dense packed colonies. I have pictures for them as well. They look exactly like yeast colonies, totally non-motile and totally flat without any sign of instability.
I give you my comment.
Yeah. Oh yeah. Oh.
if we have the time, I have missed it. Unfortunately, after Terry. So I'm just curious. Do you have a simple scaling argument for the square root of time? Like, is it I could imagine that the nutrient flux simply decays like 1 over square root of time because of.
Well, so again,
yes. So therefore, the production.
Yeah. So thanks for the question. Right? So the T to 1/2 indeed comes from the diffusion of the nutrient. There's no other way mathematically it can come from, and it comes from the diffusion of the nutrient. However, and I was trying to kind of highlight it before, it is not the same limit as if you have a colony that is sitting in one place and absorbing the nutrient. It's a new emergent regime. But the 1/2 is 100% coming from a diffusion equation. It's kind of 1/2 is the default you expect from a diffusion equation. It's kind of the Fisher equation is surprising because it gives you linear scaling in the diffusion equation, right? And so if you know, I think that's the answer.
Yeah. Thank you.
Okay, well, let's thank you.